Hello students, welcome to Distinction Learning Platform, and in this video we are talking about spermatogenesis. And before we begin this lesson, make sure that you subscribe, comment and like the video. And in the comment section you'll find a link that will take you to the website where you find other courses. Now let us begin. What is this spermatogenesis that we're talking about? Spermatogenesis basically is just the process of production of what we call sperm cells from the germ cells. So let us write that definition. So what are we simply saying? Spermatogenesis, we're saying, is just the process. So I'm just saying it's the process of sperm cell production, sperm cells production, all right, from what? From the germ cells. So now, to fully understand this definition, we need to highlight certain important terminology so that we understand fully. So what are these terminology that we're talking about? Germ cells. I like this one because it will help us to understand something. So the process of sperm cell production is what we call spermatogenesis. All right. Now, this sperm cell production, basically they come from what we call germ cells. Now, you need to understand to say these germ cells, they come from the fetal life as well as in the what? Adult life. So we need to understand the different types of germ cells that comes from the fetal life as well as in the adult life. So now we start with what? The fetal life development of the germ cells. Now we'll progress to the adult life of the germ cells. From there, then that's when we'll start talking about the formation of sperms. All right. Now, this fetal life, let us jump to week 3. So this is the week 3 structure of the fetus. So this is what we call week 3. Okay. But just to remind you to say, in week 1, can you remember what happens in week 1? Week 1, basically, that's where you can find fertilization as well as implantation of the fertilized egg. Then in week 2, what can you find? That's where now there will be the development of the embryoblast that comprises of hypoblast and the epiblast. So make sure to remember that. And in week 3, that's where now gastriation process is going to begin, where now different organs will start develop. A lot of organs in week 3 they'll start to develop. So that's when now the important organ that we need to discuss under spermatogenesis. No wonder we need to start from week 3. Okay, so now this is week 3 of the fetal life. So we need to understand important structures for all the fetus in week 3 so that it can help us to understand the germ cells. Okay, so let us look at this structure. This structure is what we call the yolk sac. This is the yolk sac. Okay, this is the yolk sac. Okay, then this structure, I'm just going to put the important structures that are going to help us understand the spermatogenesis. So another structure, as you can see, this tube. Okay, this tube is what we call allantois. Okay, that tube is what we call allantois, and we'll look at the importance of that. And this is basically the stomach. Okay, like that. And this is now, let us use red for this one because it is an important structure. Okay, this one. Now, this one is what we call gonadal ridge. So let us use black. Gonadal ridge. This one is what we call gonadal ridge. Or basically just a ridges. So let's just call it gonadal ridge. And this gonadal ridge, that's where now the mature gonads are going to come from. So we need to know it. And this part is basically the sacral region, and this part basically is the one that will develop into the head of the fetus. All right. So now, let us look at what happens. Where can you find these germ cells in the fetus? Where can you find them? And what is the special name for those germ cells? Because we've got a lot of germ cells. What is the special name for that germ cells? We call them primordial germ cells. So let us name them. So the type of germ cells that can be found inside the fetus or fetal life, we call them primordial germ cells. What do we call them? We call them primordial germ cells. Sometimes they are abbreviated as PGC, primordial germ cells. That is the germ cells that can be found in the fetal life. Now, the question that you can ask is that where exactly can we find these primordial germ cells in week 3 of the fetal life? Where can you find them? Basically, they are located in the yolk sac. Okay, these are the primordial germ cells. Where are they located? They are located in the yolk sac. But basically, if you look at the yolk sac, it is a big structure. So we need to specify exactly where in the yolk sac you can find the primordial germ cells. So these primordial germ cells, although they can be found inside the yolk sac, but they are very near to an important structure which we call allantois structure. That allantois structure, basically, it is an important structure in the development of the hindigut. We'll come and talk about the development of hindigut in future. So, but basically, just remember to say allantois structure, it is an important structure that participates in the development of the hindgut. Alright? Now, we are sent to say these primordial germ cells, they are very near to the allantois, basically. And what happens is this. Since we know the location of these primordial germ cells, basically they can be found where? They can be found just near the allantois structure, and they can be found in the yolk sac. Alright? Now the question that you can ask is that, how do they find themselves in the gonadoid? Because here in the gonadoid, they need to be there so that now we can have the development of what? Now the gonads, the male gonads. Okay? So how do they find themselves there? So they will start migrating because of some various signals that are going to be happening in the fetus. Those primordial germ cells, they will start migrating from this side up to the gonadal regions. Now they'll follow a certain structure that I'm going to name. So they'll start moving like this. You'll start moving like that, like that, like that, like that, like that, like that, like up until they migrate into this gonadal ridge. So this path they take is what we call, I need to write this one. So this path that they take is what we call mesentery, dorsal mesentery, sorry, we call it dorsal mesentery of the hindigut. Okay, you need to take note of that. Okay, dorsal mesentery of the hindigut. Okay, so take note of that. So dorsal mesentery of the hindigut, that's where it's a pathway, or it is the way in which these primordial germ cells, they use to migrate from the yolk sac up to the gonadal regions. All right, but why am I troubling you with the migration of these primordial germ cells? It is because there are some things that can make sure that these primordial germ cells can't migrate well and they can't reach these gonadal regions. If that happens, basically that person is going to develop, or that fetal is going to develop some abnormalities like cancer of those primordial gonadal cells, because they like to divide. So if they are not found in the gonadal they are found outside, basically they can develop what we call cancer. So we don't need that. Basically, they need to move from the yolk sac up to the gonadal regions. All right. So that's why it's very important for these to migrate to prevent cancer. All right. Now, since we've understood to say they are here, and this migration, the migration of this, it happens in what? So let me write it. It happens in between week 4 to week 6. So take note of that. So the migration of these primordial germ cells happens between week 4 to week 6. So make sure that you understand on that, all right? Okay? Now, we need to understand the next step now. What happens to these gonadal regions? The moment these germ cells find themselves in the gonadal regions, what's going to happen to these gonadal regions? Something very fine is going to happen. So you know what happens is that these gonadal regions, they have that ability to differentiate or to change into the male gonads and the female gonads. Okay, so what are we simply saying? What we're simply saying is that these gonadal regions, they have that ability to change into what? The female gonads and the male gonads. Okay? Yes. So we need to understand something like that, okay? Again. So these gonadal regions, although they are receiving these germ cells or the primordial germ cells, but they've got that ability to change into the female gonads or the male gonads. But the question that you can ask is that since we're talking about spermatogenesis, how do they know that we need to develop into the male gonads? How do they know? Okay, and this is the interesting part. So hopefully you've taken note of this. Okay. So how do they know? That's the question that you need to ask. How do they know that we need to develop into the male gonads? If I take you back, we know that if I represent something like this, then genetically speaking, that is a female, XX chromosome, that is a female, and XY, this is what? A male. All right, we know that. Now, since these gonads that are coming into the gonadal ridge, they are carrying this XY chromosome. Okay, why do I say that? Because after fertilization, for us to be talking about spermatogenesis, we know that after fertilization, the embryo is carrying XY chromosome. So along these germ cells that are coming here, they are also having XY chromosome. Okay, so now this XY chromosome, the very important component is the Y chromosome. The Y chromosome is the one that will go to the gonadal ridge and tell the gonadal ridge to start developing into what we call the male gonads. That's how it's going to know that we need to now develop into what we call the male gonads. So pay attention to this. Again, what we're simply saying is that these primordial germ cells, okay, let's just say PG, primordial germ cells, they carry the Y chromosome. They carry what? The Y chromosome. And the Y chromosome is the one that's going to stimulate what? The gonadal ridges to start developing into the male gonads. All right? Now, there is an important thing that you need to understand as well. All right? Now, since we're saying the Y chromosome is one that's going to stimulate the gonadal ridges to start developing into the male gonads. But if you look at how they develop into the male gonads, we need to understand something here. How do they develop into the male gonads? So these gonadal ridges, they've got two important cells. So these gonadal ridges, they are going to start developing two important cells. Okay, if they are stimulated, so what's going to happen is this. Since they are stimulated by the primordial germ cells, okay, they'll start differentiating or producing two important cells. Okay, they will start producing what we call fetal Satori cells. So let's just say Satori cells. So make sure that you follow me. Fetal Satori cells as well as fetal Leydig cells. Leydig cells. Okay, so two important cells that are going to be developed after they are stimulated by the Y chromosome. Okay, they are going to have the fetal Satori cell. So this is Satori, that is the Satori cells, fetal Satori cells, and the fetal Leydig cells. Fetal Satori cells, what do they do? These fetal Satori cells, make sure that you understand this because they are going to examine on that. So fetal Satori cells, what do? They will start producing something. Okay, so these fetal Satori cells, they produce something which we call Ant-Muranian hormone. Okay, Ant-Muranian hormone. This one, let me write it in full here. Ant-Muranian hormone. Okay, Ant-Muranian hormone. So this Ant-Muranian hormone, abbreviated as AMH, they are going to prevent what? They are going to prevent they are going to promote what we call the regression of the Muradian ducts. Okay, so they promote the regression of what? The Muradian ducts. So let us write something here. So this, what's the function of this? Promotes regression of Muradian ducts. Okay, so make sure that you take note of that. Okay, so what is the function of that hormone that is going to be produced by the Satori cells or fetal Satori cells? They are going to produce what we call Ant-Muranian hormone that's going to promote the regression or the disappearing of the Muradian duct. Why is it important? Because Muradian ducts, if you remember, they are important in the development of what? The uterus, as well as the upper vagina, as well as the oviduct. And we don't need those structures in the male reproductive system. No wonder they need to regress. So this hormone is very important. Okay, again, to take you back, we're saying the primordial germ cells, the moment they go into gonadal, they're going to produce this Y chromosome is going to stimulate the gonadal to start developing the fetal Satori cells. And these fetal Satori cells, they will start producing this hormone, which we call Ant-Muranian hormone. And this hormone is going to start doing what? Promoting regression of what? The Muradian ducts. Why? Because we don't need the female reproductive structures in the male reproductive system, like what? The oviduct, the uterus, as well as the upper vagina. That's why it is crucial for this. All right, now let us go to this again, another one. So apart from this, we said also these PG cells, what do? They also promote the development of what? Fetal Leydig cells. Fetal Leydig cells. Okay, so these fetal Leydig cells now, they promote what? This one is very simple. They promote the production of what? Testosterone. Testosterone. Okay, they promote just the production of testosterone. So what is the function of testosterone? Now the function of testosterone now, it will start now producing what we call it will promote the development of what we call the internal structure of the male reproductive system. Again, what is the function of testosterone or fetal testosterone produced by the fetal Leydig cells? It will promote the development of the internal structures of the male reproductive system, like what? This one. So look at this one. Let me just list them. The first one is what? Vas deferens. So you can say vas deferens, which is the sperm duct, vas deferens. Okay. The second one is what? Is promote the development of epididymis. Didymis. Okay. Number 3 is promote the development of seminovesicles. Okay. The last one is promote the production of, or the development of, seminiferous tubules. Okay. So now again, you've seen these internal structures. So we are saying the fetal Leydig cells, they promote now the development of the vas deferens, the epididymis, seminovesicles, seminiferous tubules, as well as now other important structures. Okay, that's the function of testosterone. Now, you can also ask to say, what about the external important structures of the male reproductive system? Where do they come from? So those external structures, like the penis, okay, the urethra, those, and also the testes, they come from what we call also testosterone, but this testosterone has to be converted into something which is very important, which we call the dihydrotestosterone. So this testosterone, for it to now start promoting the development of external structures like the penis, the testes, it has to be converted into dihydrotestosterone. Okay, so maybe you can write that one. So I move this. So what you are simply saying is the testosterone, this testosterone, so let's say testosterone has to be converted into dihydrotestosterone for it to what? Promote the development of male reproductive structures. Male reproductive structures. Like what? The penis, okay, maybe we say the testes, okay, what else? Maybe the prostate gland, okay, prostate gland, etc. Okay, and many more. So these are the things that you need to understand. Now, since we've understood to say, so in summary, so let us summarize whatever we've talked about. Now, what you've talked about is this. So we said to say, quite all right, the germ cells. These germ cells in the fetal life, around week 3, there will be the germ cells that will be developing in the yolk sac, which we call primordial germ cells, PGC, okay? That will develop in the yolk sac near the allantois structures. And around week 4 to week 6, they will start migrating from the yolk sac up to the what? Gonadal ridge via a certain structure which we said dorsal mesentery of the hindgut. That's what we said. Now, upon reaching the gonadal ridge, what did we say? Then this gonadal ridge, it's able to differentiate or to start developing into the female gonads and the male gonad. Now that is termed what we call indifferent gonadal ridge. Okay, so now it's able to differentiate into the female gonads and the male gonads. Now what happens is that because these primordial germ cells that are coming, they are carrying the Y chromosome. The Y chromosome will start now sending signals to these gonadal ridges to start now differentiating into the male gonads. But the question that I asked is that how do they differentiate into the male gonads? Okay, it's because of what? this PG or the Y chromosomes is going to stimulate this gonadotroph to start developing fetal Satori cells. Fetal Satori cells, they produce this hormone which we call anti-Müllerian hormone, that's going to promote the regression of Müllerian duct, because Müllerian duct promote what we call the formation of oviduct, the formation of the uterus, as well as the upper vagina, and we don't need those structures in the male reproductive system. One of those structures has to break down. The next, also, there will be fetal Leydig cells that promote the testosterone production. And testosterone production, it has two important functions, which is promotion of what? Internal structures, development of the male reproductive system, as well as the external male reproductive system development. All right. And what are the internal ones? It's vast difference: epididymis, seminovesicous, seminiferous tubules. Now the external one are penis, testis, prostate gland. All right. Now let us move. Okay. Now this is the structure of the internal structure of the testis. Okay. So we need to understand it as well. So if you look at this structure, it has important structures that are going to help us to understand about spermatogenesis. All right, what are those? We've got this structure here inside. These black structures, we call them seminiferous tubules. So what we call this? We call these as seminiferous tubules. Okay. And we call this part, this whole part, we call it the head of the epididymis. This is the head of the epididymis, and this is the body, this part. This is the body of the epididymis, and this is what? The tail of the epididymis. All right. So the head, body, and the tail of the epididymis. And this whole part is the vas deferens. All right. So those are the important structures that we need to understand. Now, what we need to understand is that these seminiferous tubules are very important in what? Spermatogenesis. So what we're going to do is we're going to get a cross section. So we're going to get a cross section of this seminiferous tubule. And if you get this cross section of the seminiferous tubule, what we're going to see is this. Okay, so this is what we're going to see from the cross section of the seminiferous tubule. Why are we doing that? So that we fully understand how the sperms are going to be made. All right. So now let us begin with that. So now, here we talked about the migration of the primordial germ cells. Okay, primordial germ cells, that's what we talked about. So the primordial germ cells, they are going to find themselves in this structure which we are saying seminiferous tubule. So the primordial germ cells, they are going to be found themselves in the what? The seminiferous tubules. Now the moment they are in the seminiferous tubule, they are going to change their name. So we're going to change the name. Okay, since now what we need to understand is this. Okay, we need to understand that fetal life and adult life. Okay, so let us understand something. So in the fetal life, the type of germ cells that we're talking about is primordial germ cells. But after birth, the primordial germ cells, they are going to change or differentiate into something which is important. Okay, and in adult life or after birth, let's just say after birth, these primordial cells, they're going to change into what we call spermato. So it's a longer name, spermatogonia. Okay, so you need to understand this. So in fetal life, those primordial germ cells that came into this structure, We call them primordial germ cells, but after birth, the name is going to change into spermatogonia. Okay? Now, since we've understood that, now let us try to understand this structure, the names of this structure. Then after that, we start talking about what happens. Alright? So, we've got this shady thing here. This is a capillary. Okay? This is a capillary. Now, we've got these important cells. Okay? What do we call these important cells? We call these as, now we had fetal Satori cells. Now, these are what we call adult Satori cells. Now let's just say Satori cells. So these are what we call Satori cells. Okay. So Satori cells, they can be found just inside the seminiferous tubules. Okay. Now in the connective tissue, we've got this structure. Okay. This structure within the connective tissue of the what? Seminiferous tubule. We've got these cells again. What we call these cells? We know now these are Leydig cells. Leydig cells. And in the fetal life, we had fetal Leydig cells, and these are what? Adult Leydig cells. Alright? Now, apart from that, we've got this structure. You know that in capillary, they are going to have the basement membrane. So, this black thing is what we call the basement membrane. Okay. Yeah. So these are the important structures that you just have to remember. The basement membrane, okay, the capillary, these structures which we call Satori cells, and these cells which we call Leydig cells. Alright. Then the other important thing is that where can we find the spermatogonia or the primordial germ cells? Okay, where can you find them? We can just find them just after the basement membrane. Where can you find them? You can just find them here, somewhere here. They are a lot here. Okay? They are here. These are what we call what? We call them the spermatogonia after the basement membrane. That's why you can find them. Now, the question that you can ask is, these spermatogonia, what are they going to start doing? So these spermatogonia, they are going to be stimulated by something. Okay. So before anything happens, they don't just start proliferation and differentiation into other cells by their own. On their own, they are going to be stimulated by something which we call the pituitary gland. So let us write the pituitary gland. I'm going to remove this. Okay. So before I start anything there, we need to understand to say these, they are not going to start working minus the stimulus. Okay, they receive certain stimulus from the pituitary gland, right? All right, so that's the pituitary gland. So if you just remember to say the pituitary gland is connected with what? The hypothalamus, and this is the pituitary gland. This is what we call the pituitary gland. So now, hypothalamus is going to stimulate the pituitary gland, but basically we're only interested with the anterior pituitary gland. Okay, but this is the posterior. Remember that the pituitary gland is divided into the anterior pituitary gland and the posterior pituitary gland. The hypothalamus is going to start sending certain hormones, okay, or gonadotrophin, releasing hormones that are going to be produced by the hypothalamus, and they're going to stimulate the anterior pituitary gland to start producing certain hormones. Okay, two important hormones which we call luteinizing hormone and follicle stimulating hormone. Two important hormones, luteinizing hormone and the follicle stimulating hormone. Okay. Now, what's going to happen is this. Okay, so what's going to happen is this. So, this follicle stimulating hormone. So, the follicle stimulating hormone will go to these cells. Okay, what did we say these cells? We say these are what? Satori cells. Okay, Satori cells. What's the function of Satori cells? So, this now, the function of Satori cells is going to change in the what? In the adult life. Okay, remember that in the fetal life, we said it's going to produce a certain hormone which we call anti-Mullerian hormone, but here it will start producing something because this is an adult Satori cells. So we said if this is stimulated, it's going to produce some certain important substance, which is androgen binding protein. This one is a very important structure or important protein that we need to discuss. All right. Again, if you look at the luteinizing hormone, okay, this luteinizing hormone will go to the what? Will go to the Leydig cells, and these Leydig cells now, the one that can be found just outside the inside the connective tissue of these seminiferous tubules, they are going to start producing what? Certain important hormone here. So these Leydig cells are going to start producing what we call testosterone. Testosterone. Okay? So testosterone and these are going to bind together. Okay? So just think like testosterone is going to be covered by this protein, because testosterone is lipid soluble, and inside this lumen there is water, and since this is lipid soluble, it has to be soluble so that it can start stimulating now these primordial germ cells or the spermatogonia. So no wonder the androgen binding protein will surround this. Okay, it's going to surround this one so that it becomes soluble like that. Okay. Then what's going to happen if this testosterone becomes soluble? It will come here. This testosterone will come here and stimulate the spermatogonia. That's how spermatogonia now is going to be stimulated so that it will start differentiating into something that we need. Again, we go back. How do these spermatogonia, the one that came from primordial germ cells, how are they going to be stimulated so that they start differentiating into something that we want? It's because of what? The hypothalamus. Remember that this is happening in adult life, specifically after puberty. That's how when this is going to start happening. The sperm production will start happening now in after puberty in males. Alright? This hypothalamus, remember, hypothalamus is going to be stimulated, and it's going to stimulate the anterior pituitary gland that will produce two important hormones: the follicle stimulating hormone and luteinizing hormone. Follicle stimulating hormone will go to these cells, which we call Satori cells. Satori cells, what do? They produce this androgen binding protein. All right. Then again, what about luteinizing hormone? They will go to the Leydig cells that will produce now testosterone hormone. Now, the reason why this testosterone has to be binded by this protein that was produced by the Satori cells, because testosterone is what? Lipid soluble. And inside this lumen, there is the fluid, alright? And this testosterone has to dissolve in that fluid. So what's going to happen is that this androgen binding protein will make sure that testosterone is now soluble and concentrated in that lumen. After that happens, this will go to the what? Spermatogonia, and stimulate the spermatogonia to start now differentiating into something that we need. Alright? Now let's continue. So since this has been stimulated, this is the spermatogonia with now 46 chromosomes. So remember that spermatogonia, it has what? 46 chromosomes. Okay? So what's going to happen is that this spermatogonia is going to differentiate or divide into two important cells. So because spermatogonia, we've got type A spermatogonia and type B spermatogonia. So we've got A and B. And both A and B, they have got 46 chromosomes. So make sure that you understand about that. Okay? So what's going to happen is that type A, okay? Type A will go back there in that base after the basement membrane, so that now we can replenish or we can have more of spermatogonia. So type A, that helps in the what? Like proliferation of more spermatogonia. So it will go back there so that it can now replace the cells that have been differentiated into what? Type B. Make sure that you understand about that. Okay? Then type B, it will start now differentiating into things that we need. Alright? So type B spermatogonia will start now dividing. Okay? We'll divide into these two. Okay? So it will differentiate into that. So let us just write one. So type B will divide into this cell, okay, which we call the primary spermatocyte. So it's going to develop into what we call the primary. So let's just use this primary spermatocyte. That's the first thing. Primary spermatocyte by what we call mitosis. Okay. So primary spermatocyte, and you need to understand to say primary spermatocyte is going to have about 46 chromosomes. So that's the first thing. The type B spermatogonia or spermatogonium, they're going to divide by mitosis into primary spermatocyte, but it's going to have 46 chromosomes. So what's going to happen here is that, so we make more space here. Okay, this one is going to divide by mitosis. Okay, it's going to divide by, meiosis 1, okay, into secondary spermatocytes. Okay, again, what we're simply saying is that this primary spermatocyte is going to divide by meiosis now. Meiosis what? Meiosis 1 into secondary spermatocyte. Now you need to take note of something here. Okay? This one is going to have about, here we had 46, but here we're going to have 23. Okay? Make sure that you understand. Primary spermatocyte, they've got 46 chromosomes. Secondary spermatocyte, they're going to have 23 chromosomes. Okay? Now what's going to happen again? These two cells are going to further divide into what? Meiosis. They're going to divide into something that we need here. So these two, this one and that one is going to divide. Also this one and also that one is going to divide as well. But both with 23. And these are called spermatids. Okay? Spermatids, they are going to do, so this one, it is in meiosis 2. Okay? So we need to understand again, the secondary spermatocytes, they are going to divide by meiosis 2 into spermatids. Okay? So, and these spermatids, again, they are going to have about 23 chromosomes. How many spermatids that they are going to be produced? We're going to have about 4 spermatids. So, we're going to say one primary spermatocyte is going to produce about 4 spermatids. Okay? Now, these 4 spermatids, they are going to further differentiate. Now there will be no division up to here. So up to here there will be no division. Okay. From here up to here there was division. Then from here up to the stage that we're going to talk about, there is no division, there is just differentiation. So these spermatids, so these are spermatids as well. These two, they are also spermatids. Spermatids. Okay. So these spermatids now, they are going to differentiate into something. Okay. These spermatids, they are going to differentiate into what we call now, okay, spermatozoa. Spermatozoa. Okay. They are going to differentiate into spermatozoa by the process called spermatogenesis. Spermatogenesis. Okay, so you need to understand something. The change from spermatids up to spermatozoa is what we call spermatogenesis, because now the spermatids, they will start now what we call like they will start maturing into something that we need, the mature spermatozoa. Okay, so this is the whole process that's going to happen in the what? In the seminiferous tubule. Okay, and another important thing is that you need to remember to say these mature spermatozoa, okay, they've got that ability to produce maybe antigens. Okay, now these spermatozoa, since they've got that ability to produce those antigens, they are not, but however, they are not attacked by what we call antibodies of the body. Reason being is that because these Satori cells, they've got those tight junctions, so the antigen, they can't go into the blood or the circulation there, so that they can trigger the immune system to start attacking these spermatozoa or the mature cells. All right. So again, the reason why the sperm cells are not destroyed in the testis there is because of the Satori cell. The Satori cells have got these tight junctions that are this tight junction between the cells. So this cell and this cell is connected between the what? The tight junction. And this process is happening between the Satori cells. And this tight junction, these are the ones that are going to form what we call the blood testis barrier. So there will be blood testis barrier because of this what? Tight junction. No wonder if you look at this tight junction, they're going to divide this funiculus tuber into what? Two important compartments: the basement compartment or the basal compartment and the tubular compartment. Okay, the basal compartment and the tubular compartment. You need to understand, okay, about the tight junction. Now, the last important thing that we'll talk about is this one. So this spermatozoa, the structure of the spermatozoa, is going to have the head. It's going to have the head, and inside the head, we're going to have two important structures that you need to remember. We've got the nucleus, okay, the nucleus, which is condensed, the nucleus, and this nucleus is going to have an important structure here. So this nucleus is going to have this structure in black anterior. So just anterior to the nucleus, we're going to have this important structure which we call acrosome. Why acrosome is important? Because it contains enzymes that are going to help in fertilization or the penetration of the zona pellucidum of the ovule. Okay, that's why it's important, the acrosome. Okay, then we are going to have the neck. Okay, and below the neck we're going to have a certain important structure here. Okay, a certain important structure that contains a lot of mitochondria here. Okay, and we've got the tail. Okay, or sometimes it's referred to as flagellum. Okay, the flagellum is important in the what? Movement of the entire sperm cell. And on the neck, you need to have this structure, okay, which is also important in what? Production of ATP. Okay, on the neck we've got that structure that is also important in the what? In the ATP production, so that now the sperm cell can move from what? From what we call from one place to another, right? And this is where we end from. All the best until next time.